Hello, everyone. My name is Laura Nilsson, and I'm the Associate Dean for Graduate Education in the Faculty of Science. And on behalf of the Dean and the entire Faculty of Science, we would like to welcome you today to the 10th Sun and Science session, which is the summer edition of our regular Soup and Science series. Today we have three incredible speakers who will be presenting about their research. We have Professor Samantha Gruenheid from the Department of Microbiology and Immunology, Professor Alvin Shrier from the Department of Physiology, and Professor Don Sheppard, also from the Department of Microbiology and Immunology. Once their presentations have been completed, you will each receive a message from the host to join a breakout room. In this breakout room, you will be in smaller groups, and you'll have the opportunity to ask questions to each presenter and discuss a little bit about their work. Each presenter will rotate through the rooms every 10 minutes until they've been in all three rooms. And at that point, we will all head back to the main room for final remarks. So thanks everyone for coming, and we'll begin with Samantha's presentation. Go ahead, Samantha.
Okay, good morning, everyone. It's a pleasure to be here, to be presenting to you, and I'm going to be talking to you about a project that did in collaboration as part of a research team with two professors at McGill and two professors at University of Montreal. And before I start, I just want to say that most of the work in my lab on this project was done by Tyler Cannon, who is someone that joined my lab in his U1 year, after his U1 year for the summer, and he stayed on and he's completing his PhD on this project right now. So in this multidisciplinary project, we investigated the links between the gut and the brain in Parkinson's disease. So Parkinson's disease is a common chronic progressive neurodegenerative disorder that is described with the development of motor symptoms such as tremors, stiffness, and difficulty walking, and that's associated with the loss of a specific set of neurons in a specific region of the brain, so the dopamine-producing neurons in the substantia nigra, and that's shown here. So this is a top view of your brain. This is this little butterfly-shaped structure, and the substantia nigra is here. And it's a very complex disease, meaning that it's influenced by both genetic and environmental factors. And what really sparked my interest as someone that studies the intestine and intestinal infection and inflammation is that ever since Parkinson himself described the disease in 1817, there was this implication of the intestine influencing the brain in this disease. So Parkinson noted that there was gastrointestinal symptoms such as constipation that precedes the development of motor symptoms in about 50% of Parkinson's patients. And also, there was a natural experiment where before people knew that Helicobacter pylori was the cause of ulcers, people thought that they were caused by this vagus nerve, which actually connects the brain to the gut. And so to get rid of ulcers, they tried severing the vagus nerve. And what was found later is that people that had their vagus nerve severed for this reason were at 50% decreased risk for later developing Parkinson's disease in their lifetime. And in addition, recent studies have looked at the same thing with people that have had their appendix taken out, who are also at lower risk for developing Parkinson's disease. So this was more recently supplemented with new data where people have gone back and looked at the online health records of hundreds of thousands of people and seen that people that have seen their doctors for inflammatory bowel disease, which is a disease, a chronic disease of intestinal inflammation, are also at increased risk for Parkinson's disease over time. And that's just shown here with the incidence of Parkinson's disease over time. And you can see that the inflammatory bowel disease patients have a higher incidence than the control group, which would be the same age and matched as well as possible to the patients. In addition, a similar study was done looking at people that had serious gastrointestinal infections who were also found to be at increased risk of Parkinson's disease. So these were people that had a serious enough gut infection that they would have seen their doctor and had this in their medical chart. And so because people didn't really know any of the mechanisms connecting this intestinal inflammation and disease and this brain and motor phenotype, we wondered if it would be useful to try and model this in mice. And what we thought was very interesting is there's a gene called PINK1 that's very highly implicated in Parkinson's disease in humans. So if you're unlucky enough to inherit 2 mutations in PINK1 from your parents, so a homozygous mutation, you have about a 95% chance of developing Parkinson's disease. So this is quite rare, not to worry, but also people that are homozygous for PINK1 have an early onset disease, so have a higher chance for early onset disease. So this is a disease that we think about developing later in life, but people with PINK1 mutations can get it in their 30s and 40s. However, if you make a mouse that's genetically engineered to have 2 mutations, homozygous mutations in PINK1, there's no symptoms related to Parkinson's disease and no loss of these dopamine-producing neurons. So thinking about that, we realized that humans are continuously exposed to infectious stimuli and have many minor infections throughout their lifetime. However, what you might not know is that lab mice, when we keep them for experiments, they're kept in a very protected environment that keeps them free of exposure to these infectious pathogens. And so what we decided to do is infect these mice with a natural pathogen of mouse known as Citrobacter rodentium. So this is a bacterial infection that colonizes the intestinal tract of mice and leads to a self-limiting disease. So there's an increase in bacterial loads and then a clearance. And at the peak of infection, mice have sort of gastrointestinal symptoms, but then they go on and recover. And what we found quite strikingly was that not during the infection, but several months following infection, the PINK1 mutated mice, oops, I have to go back. The PINK1 mutated mice developed these symptoms of Parkinson's disease. So that's shown in this movie. This is 4 months following infection. You can see on the right-hand side that the mice that had previously been infected developed symptoms such as less movement of their hind paws, less vertical episodes, which is when they stand up on their hind legs, and splaying and an altered gait. So we went on to use a way that scientists can actually measure how much the motor symptoms are affecting the mice. And this is called the pole test, where you place a mouse on top of a pole and time how long it takes to turn around and descend the pole. And what you can see here, this is a PINK1 mutated mouse that has been previously infected 6 months ago. And this mouse has been injected with L-DOPA, which is a drug that's used to treat Parkinson's disease and can be converted to dopamine. So it restores the function of the dopaminergic system in the brain. So you can see that that mouse descended the pole really quickly. This mouse hasn't quite figured out how to do it. In the interest of time, I'm going to skip ahead. And you can see that this mouse really takes a much longer time to descend the pole. And when it does descend the pole, it's having a bit of trouble coordinating its motor symptoms. So in the interest of time, that's all I'm going to talk about. I'm happy to talk about it more in the breakout rooms after the session. But in this project, we've developed a new model to study this role of intestinal infection and inflammation in Parkinson's disease. And we're using this to uncover new mechanisms that people didn't previously know were involved in the development or the triggering of disease. And we hope that these findings may help us develop new interventions that would slow the development of disease in people that are already genetically susceptible to Parkinson's. The last slide. Yeah, here's the acknowledgements. So this is Tyler, who I said joined my lab in the summer after U1 and is currently completing his PhD. This is the rest of my lab and the collaborators on this project. So thank you for listening, and I'd be happy to answer any questions in the breakout rooms.
Thank you. Thank you very much, Samantha. And our next speaker will be Alvin Shrier. Go ahead, Alvin.
Okay, I don't have the slides yet, but
Uh,
we need to shift over to my slides. There we go.
Alvin, you have controls.
Okay. So now I have to go to, okay. Well, it's great to be here today, to have a chance to connect with you all, and a special hi to Samantha and Don, my colleagues in the new School of Biomedical Sciences, where it's exciting opportunities for undergrads to do some research. There you go. Also, I'm a professor in the Department of Physiology. It's located up in the McIntyre building, but my lab is down below here in the Bellini building, where I direct a group called the Cell Information Systems Group, where our focus is on membrane proteins and involved in ion channels, transporters, and receptors, which are implicated in various diseases related to neuronal systems, cystic fibrosis, and cardiac arrhythmias. I'm going to tell you today about some work in my lab related to a condition known as long QT syndrome. Long QT syndrome is a prolongation of the QT interval of the ECG represented in this green area. This prolongation of the QT interval under stressful conditions predisposes you to arrhythmias called torsades de pointes, which can lead to fainting, seizures, and worst of all, sudden death. Now, there are at least 12, maybe now 14 genes that have been implicated in long QT syndrome. I'm going to tell you about one of them that I'm working on. It's called HERG, H-E-R-G, human ether go-go related gene. This encodes for a potassium channel called HERG that has a current called IKR. IKR is an outward movement of potassium across the cell membrane, which leads to the repolarization of the cardiac action potential. So on top, we have a picture of a cardiac action potential, which is an area of excitation, which occurs with every cycle of your heart. And when the action potential duration is prolonged in the ventricle, it causes the prolongation of the QT interval on the ECG. Now, this can be, in the case of HERG, inherited, and there are 400 different mutations, or more than 400 mutations in HERG, and it occurs in about 1 of every 2,500 people in the population. But it also can be acquired, because HERG is very promiscuous. It likes to bind drugs, and because of that, the Federal Drug Administration in this stage requires that all new compounds are screened against HERG, not to block HERG channel. Now, when you have mutations in HERG, there's protection of the cell so these mutated channels don't get to the cell membrane. Well, in that, the first level of quality control is ER, endoplasmic reticulum quality control, which takes these mutated proteins that are misfolded and sends them for degradation. But some of these channels that are not so severely mutated can actually escape the quality control, and they can go to the cell membrane up here, where they encounter a second quality control, a peripheral quality control, which is something we found about 5 years ago. Now, we looked into this peripheral quality control, and we found that these mutated channels that are not so severe, and they occur in a certain domain called the pass domain of the channel, when they get to the cell membrane, they get internalized more rapidly, they get recycled less quickly, and they get degraded more quickly. This leads to a reduction of the potassium channels at the cell membrane and the prolongation of the action potential. We don't know the mechanisms involved, and it's something that we're looking into right now. However, if we could counter these defects, maybe we could rescue the phenotype, and then even with those channels that make it, we can keep them stable and then keep the action potential duration from prolonging. So we did a screen of a library of 5,000 compounds, and we found that 153 of these compounds could actually increase the stability of the channel at the cell membrane. However, we did a second screen to make sure they don't block the channel or interfere in function, and we ended up with 15 candidates. And from further screening, we went from 15 to 5 rescuers, and finally 2 correctors. These 2 correctors are anagrelide, which is a drug used for platelet reduction, and the second compound, DC Bio, which is an activator of potassium channels. Now, at this point, we started investigating how is this actually happening. What we found was that these stabilizers, so to speak, reduced the rapid internalization into the cell, and they also reduced the rate of degradation of the mutated proteins. Again, we don't understand the mechanism of how the drugs are working at either level, something that we're also looking into. But one thing we did know that these two drugs are not additive, so they work by the same mechanism, and we're working with a chemist to develop analogs that could be more specific and more targeted to try and get a better rescue. Just to finish off, I want to say that this work is done in collaboration with my colleague Gergely Lukács, and primarily by a PhD student who's now a postdoc in Germany, Brian Fu. And with that, I'm going to stop and thank you all very much.
Fantastic. Thanks to you, Al. Our final speaker before the breakout rooms will be Don Sheppard. Over to you, Don.
That worked well. I didn't have to restart mine, unlike the other ones. Something's fixed in Zoom. So I'm going to give you a broad overview of our program and why we study what we study, and I'm going to tell you about a thread that runs through this particular program. So I'm a clinician scientist, and what I do in the lab is really driven by what I see in the clinic. And what I see in the clinic is a very odd kind of clinical practice. I actually deal with human fungal diseases, which seems like a bit of a weird career choice. I can assure you most people feel that way. But what you have to understand is that fungi are not really a big problem in healthy people, but if you're immunocompromised, they can be disastrous. So every day of your life that you're walking down the street, you're inhaling airborne spores of fungi. And it's estimated that you inhale somewhere between 100 and 1,000 spores of my favorite fungus, Aspergillus fumigatus, every day of your life. And if you're in a moldy old building like most of McGill's buildings, that could be as high as 10,000 spores a day. What happens, of course, is you have a very effective immune system that actually kills those spores or eliminates them mechanically from your lungs before they get a chance to cause any trouble. Either the little cilia, those little fingerings that are inside your lung, wave them all the way up and you spit them out, or you have white blood cells that come in, phagocytize them, and kill them. And these spores just sit there like little inert balls and do nothing. But modern medicine is amazing at screwing up your immune system. We do it on a daily basis to treat your cancer, to give you a bone marrow transplant, an organ transplant, to treat your inflammatory diseases like arthritis. And when we do that, and when we mess up your immune system, we impair your immune system's ability to defend against this constant daily onslaught of fungal spores. And what Aspergillus does, if it doesn't get killed, is it acts like a seed in your lung that germinates and forms a filamentous protuberance called a hyphae, which is, if you think of it, just like the germ tube that comes out of a seed. And that filament invades into your lungs, and it produces digestive enzymes that literally digest your lungs, and the fungus will eat you from the inside out. It'll degrade and destroy your lung tissue until it hits something important like a blood vessel, at which case you will then drown in your own blood and die. And this is what happens in up to 20% of transplant patients. It's as horrific as it sounds. And so I've been fascinated with trying to figure out ways to combat these very dramatic and devastating infections. And they're very challenging to treat because fungi, if you look at the tree of life, are actually very closely related to you and I. They're eukaryotes, and they're actually more closely related to us than plants are, and way more closely related to us than they are to bacteria. So generating molecules that kill fungi but don't kill humans is the big challenge. So we think we found one of the Achilles heels of fungi, and that is that they make a thick, sugar-rich cell wall. And they produce these very complicated sugars called polysaccharides, which are unique to fungi. You and I don't have a cell wall. So they're obviously something that if we can target is very, very specific to fungi and is very, very different from humans or animals that get fungal infections. So my lab uses a variety of techniques that range from cell biology, animal models, genetic and molecular biology, and some very high-tech glycobiology and structural biology with our collaborators to identify these polysaccharides that are unique to fungi, that play an important role in causing infection, and that we can then target as therapeutics. And so the example I like to talk about is this guy here in the center, galactosaminogalactam, which is a jawbreaker, so we call it GAG. So GAG is a long linear sugar or polysaccharide that is made by Aspergillus, and it's this beautiful little filamentous stuff that you see around these thick finger-like hyphae. And it turns out that GAG is actually a gluey, sticky sugar that the fungus uses to attach to our cells and to cover itself in an armor so that the white blood cells that come in to kill it, or the antifungals that we prescribe, can't reach their targets inside the fungus. So it's a dual purpose molecule. It's both a defense and it's an effector because it allows it to stick. So what we have done using the tools we have is figure out how this sugar is made and finding all of the enzymes that are involved in the pathways to produce GAG. And of course, our initial thought was we'd find some critical enzyme that we could go in with a small molecule, just as Dr. Shrier showed you, do a nice large high throughput screen, find some beautiful inhibitors, and boom, we have a new antibiotic. In this case, antifungal. But what we actually found early on were these green fellows here, which are what we call hydrolase molecules. And these hydrolase molecules are actually enzymes that can cut the polymer. And at first you think, okay, but if I'm making this long sugar, why do I want to cut it? And the obvious answer is, it's just like if you're building a house. If you're building a house and you buy a bunch of 12-foot long 2x4s, unless you have a saw to cut those 2x4s into different lengths, your house is going to be 12 feet by 12 feet by 12 feet. It's not going to have any doors and windows because everything's 12 feet. You need to be able to cut things to put them in the right place and have them as the right size. And these hydrolase enzymes cut the sugar, but they do it in a very careful, very regulated way. You see that they're down here at the cell membrane, the mature sugar's out there in the extracellular space. And so control of these hydrolases or these molecular saws is critical to their function. What we have done with our collaborators at Sick Kids, who are expert structural biologists, is we've made recombinant versions of just the business end of these molecules, the part that cuts up the sugar. We can make them in large quantities, we can make them soluble in water, and we can add them to the outside of the fungus without their controls, without their safety functions. And of course, what they do is what they're designed to do. They go in and chew up the sugar. So it's like running through that 12-foot house that I just told you about, swinging a chainsaw in the air. And if you do that enough, the house falls down around your ears, and that's exactly what happens here. The sugar gets degraded, the whole structure falls apart, it can't stick, and the antifungals get in and kill better. And so we're now taking these hydrolase enzymes, we've done them in animal models, and we're looking at their toxicity, doing things like, again, Dr. Shrier mentioned, to make sure that they don't mess up his ion channels and kill patients, and taking those into the clinic as a new therapy. And this is only one of the type of projects that we use targeting the sugars that fungi make, and that we think are a really unique place to tackle this very important health threat. And with that, I'll wrap it up. I also have to acknowledge that I don't do any of this work. The projects here that you're seeing have been mostly led by a PhD student who's left the lab, Mark Lee, and a postdoc who's just finishing up, Francois Lemeuf, who's our glycobiology guru. And then I'll be happy to talk more about these, and a whole second story about GAG, where we think we've found a completely brand new function for it in the breakout rooms. Thank you.